Welcome back for another deep dive. You ready to tackle a tough one today?
Always up for a challenge. Bring it on.
Okay, good. Because today we're diving into the world of antibiotic resistance.
Oh, a classic. And always relevant.
Exactly. So no shortage of material to dig into. And specifically, as a medical microbiologist, we're focusing on how bacteria, those little buggers, use efflux pumps to essentially evade these drugs.
Right, efflux pumps, like tiny little bouncers at the cell membrane.
Perfect analogy. Our main source for this deep dive is Chapter 11, Antibiotic Resistance via Membrane Efflux Pumps.
From Walsh and Wenswitz's book, Antibiotics, Challenges, Mechanisms, Opportunities. I'm guessing you've got a copy close at hand.
You know me too well, I do. Always like to have the source material right here. So let's jump right in. Bacteria, they've evolved these, seriously clever ways to resist antibiotics. And efflux pumps.
They're really a major player in that game.
Oh, absolutely. Think about it. These pumps are actively, pumping antibiotics out of the cell before they can even do their job.
It's like a constant battle at the border.
Exactly. And this is, a particularly big issue in gram-negative bacteria, right?
Absolutely. Contributes to their higher intrinsic resistance. It's like they have an extra layer of protection.
Makes sense with that double membrane structure, like a fortress with two walls or something.
Right, that outer membrane, it's a real hurdle for antibiotics to penetrate.
And it's not just the extra membrane, is it? I mean, gram-negative bacteria, they have other tricks up their sleeve.
Oh, for sure. They can actually modify, the size and the number of those porin proteins in their outer membrane, further restricting, what gets in and what doesn't.
So sneaky. Can you give an example, like a specific bacteria?
Well, think of Pseudomonas aeruginosa and its OPRD porin.
Right. Pseudomonas, always a tough one.
Exactly. Mutations in the gene coding for that porin, it can lead to fewer porins, narrower porins, or even a complete absence of porins.
Wow. So they're like constantly, adjusting their security measures to keep those antibiotics out?
Exactly. It's a constant evolution.
So even if an antibiotic manages to, slip past the first line of defense, those efflux pumps are there waiting to kick them right back out.
Right. It's an impressive defense system.
But these pumps, I'm guessing they're not just sitting around, waiting for an antibiotic attack.
You're right to question that. They actually have a role in the general physiology of the bacteria as well. Take, for instance, bacteria that live in the gastrointestinal tract.
Oh, right. Tough neighborhood.
They're constantly exposed to bile salts. Our bodies use those to break down fats. But these bile salts, they can be toxic to bacteria. So they use efflux pumps to expel them, ensuring their survival.
Fascinating. So they're multi-purpose, those pumps, helping the bacteria thrive in their normal environment, and then, bam, fight off threats like antibiotics too.
They're versatile, that's for sure.
Which honestly makes them even more formidable, I guess.
From a clinical perspective, definitely.
Okay, so let's dive into the different types of efflux pumps. The book mentions 5 major families: MFS, RND, SMR, MATE-E, and ABC. Can you give us like a quick tour?
Absolutely. Keep in mind, these families, they differ in their structure, their energy sources, and the range of molecules they transport, which, by the way, is part of what makes them so tricky to target.
Ah, I see.
So starting with the MFS family, major facilitator superfamily.
Kitschy name.
You find those in both gram-positive and gram-negative bacteria, and they use that proton motor force across the cell membrane to, drive the efflux of various substrates, including antibiotics.
So they're like the generalists, adaptable to different situations, different energy sources.
That's a good way to put it. Now, the R&D family, that stands for Resistance Nodulation Division.
R&D.
And they're particularly important in, you guessed it, gram-negative bacteria. They often form that inner membrane component of those complex tripartite pumps, the ones that span both membranes and the periplasm.
Oh, right, those tripartite pumps. Always found those fascinating. You're like a 3-part machine.
Exactly. It's a well-coordinated system.
Could you remind us how those systems work?
Sure. Let's use the well-studied Akerby-Tulsi system in E. coli as an example.
Great example. Everyone knows E. coli.
So you have ACRB, that's the inner membrane pump that captures the antibiotic molecule. Then there's ACRE, the periplasmic adapter protein. It acts like a bridge connecting ACRB to Tulsi, which is the outer membrane channel.
So essentially, this whole complex, it acts like a conduit, just shuttling that antibiotic from the cytoplasm all the way out of the cell.
Precisely, like a cellular relay race.
I like that. So before we move on to the other efflux pump families, maybe it's time to test our listeners' knowledge.
Ooh, a pop quiz. I like it.
All right, so here's the question. In that Akerby-Tulsi efflux pump system, what's the specific role of the Tulsi protein?
Good one.
Take a minute, think about it. We'll discuss the answer shortly.
So that was a good question about the Tulsi protein.
Yeah, really gets you thinking about that tripartite system.
Right. It forces you to visualize the whole process.
Exactly. So the Tulsi protein, it acts as that outer membrane channel.
Right. The final exit point for that antibiotic molecule.
Essential for the whole system to work properly.
Absolutely. Without it, the antibiotic would be trapped in the periplasm.
Right.
Yeah.
No escape. Okay, let's move on to some of those other efflux pump families. We talked about SMR, MAT, and ABC.
Right. A whole alphabet soup of pumps.
So what about SMR, small multi-drug resistance? Does the name give it away?
Well, they live up to their name, relatively small, typically, 4 transmembrane helices,
Okay.
and they often form dimers or higher-order oligomers to function.
So they're like the compact, efficient models?
Yeah, you could say that.
And what about their energy source? Same as MFS and RND, using that proton motive force?
That's right, proton motive force for SMR pumps, too.
Okay, got it. So how about MAT, multi-drug and toxin extrusion? Again, the name kind of says it all.
Yeah, pretty self-explanatory. They're involved in the efflux of a whole range of substrates. Antibiotics, toxins, metabolites, you name it.
So they're multi-talented.
Yeah.
Exactly, handling all sorts of tasks.
And energy source, proton motive force again?
Yep, same as SMR.
Okay. And then we have the ABC family, ATP binding cassette.
Now, those are interesting because they use ATP hydrolysis for energy.
So they're different.
Right. Unlike the others we've talked about.
So they have their own, dedicated power source.
You got it. Independent of that proton motive force.
So they're like the, self-sufficient ones.
Yeah, I like that. Equipped with their own energy reserves.
It's incredible how bacteria have developed all these different pumps, to fight off those antibiotic challenges.
It's an ongoing arms race.
For sure. Okay, before we go too much further, I think it's time for another pop quiz.
All right, hit me with it.
So here's the question. Which of those 5 efflux pump families is unique, in its use of ATP hydrolysis for energy?
Oh, that's a good one.
Take a moment. Think back to what we've discussed.
I'm sure our listener is already formulating the answer.
Hopefully. While they're thinking, I want to mention this diversity in structure and energy sources. It's one reason why these pumps are so difficult to target, right?
Absolutely. It's a real challenge for drug development. I mean, finding a single inhibitor that can effectively block all these different pumps, that's a tall order.
Yeah, like finding a universal key for a million different locks.
A great analogy.
But researchers, they're not giving up, are they? What are some of the, the current approaches?
Oh, they're definitely working on it. One approach is to, identify efflux pump inhibitors from natural sources.
Oh, interesting. Like plants or fungi.
Exactly. Those organisms have evolved all sorts of compounds that can interfere with bacterial processes.
Right, so it's like using nature's own weapons.
Exactly. Tapping into that natural arsenal.
Like it. What else?
Well, another approach is to design a synthetic molecules that specifically target, efflux pump components.
So it's a more targeted approach.
Yeah, right. They're using computational techniques to, model the structure of these pumps and pinpoint potential binding sites.
Wow. It's like designing a precision-guided missile?
Pretty much. And then there's the use of antibodies.
Oh, antibodies. I've heard about that.
Yeah, they've shown promise in targeting a wide range of disease-causing agents.
So could they be used to, block efflux pumps?
That's the idea. The research is still in the early stages, but it's definitely an area to watch.
Exciting stuff. Okay, so back to that exam question.
Right. Which efflux pump family uses ATP hydrolysis?
The answer is, of course, the ABC family.
ATP binding cassette. Remember those?
Yep. Stands out from the crowd with that unique energy source.
Exactly.
Okay, now, we focus a lot on gram-negative bacteria. But what about gram-positive bacteria? Do they have efflux pumps too?
Oh, absolutely. They might not have that complex outer membrane, but they still use efflux pumps to combat antibiotics.
Okay, so their pumps are probably simpler, right?
Typically, yes.
Yeah.
Often just single proteins embedded in the cytoplasmic membrane.
No need for those elaborate tripartite systems.
Nope. But don't underestimate them. They can still be a major headache.
Any examples of, gram-positive bacteria known for their efflux pump shenanigans?
Well, the classic example is Staphylococcus aureus. Good old Staph.
Staph aureus.
Yeah.
Always a troublemaker.
It's notorious for developing resistance to multiple antibiotics, and efflux pumps play a big part in that.
Makes sense. So what specific pumps are we talking about in staph?
Well, one well-studied one is the Nora pump. It belongs to the MFS family.
MFS.
Right, the generalists.
That's the one. And Nora, it's known to expel a variety of antibiotics, including fluoroquinolones.
Fluoroquinolones. We use those a lot to treat staph infections.
Exactly. And Nora can pump them right back out.
So even those simpler pumps, the single protein ones, they can have a real impact.
Oh, absolutely. Nora is a prime example.
Okay, before we get too deep into Staph aureus and its Efflux pump strategies, another pop quiz.
All right, bring it on. Okay, so here's the question. Name one molecule, other than antibiotics, that bacteria commonly expel using these efflux pumps.
Thinking back to those diverse roles we talked about earlier.
Exactly. We'll reveal the answer and discuss its significance, in just a bit.
So thinking back to those other roles of efflux pumps.
Right, not just fighting off antibiotics.
The answer is bile salts.
Bile salts, of course. We talked about how bacteria in the gut use those pumps to get rid of those, potentially harmful compounds.
Right. Essential for their survival.
Exactly. Okay, so back to Staphylococcus aureus. You mentioned the Nora pump and how it plays a big role in fluoroquinolone resistance.
Right. Nora, a key player.
But are there other pumps in staph we should be aware of?
Oh, Staph aureus. It's got a whole arsenal of efflux pumps. Besides Nora, there's Nor-B and Nor-C.
More Nora pumps?
Yeah. And those contribute to fluoroquinolone resistance too. And then there are others that, expel different classes of antibiotics.
It's like this bacterium is, constantly coming up with new ways to outsmart us.
It's a constant evolutionary battle.
Makes you wonder if we're fighting a losing battle.
It's definitely a challenge, but researchers are working on it, developing strategies to overcome this efflux pump-mediated resistance.
Right, those solutions we talked about earlier, natural inhibitors, synthetic molecules, antibodies.
Exactly. There's hope on the horizon.
That's good to hear. Okay, ready for one more pop quiz?
Hit me.
All right. True or false? All efflux pumps are specifically dedicated to expelling antibiotics.
Ooh, that's a tricky one.
Think back to what we discussed, all those different roles.
Right, the bigger picture.
Exactly. While our listener ponders that, I want to ask, with all this diversity and complexity, can we really develop a universal inhibitor? Or are we going to need a specific one for each type of pump?
That's the million-dollar question. A universal inhibitor would be amazing, but it's a tough goal.
I can imagine.
The differences between these pump families, it makes it unlikely that one molecule could effectively block them all.
So maybe a more realistic approach is to develop a panel of inhibitors, each targeting a specific pump or family.
That's probably more likely. Researchers are looking at different strategies, targeting binding sites, disrupting assembly, interfering with energy sources.
So many possibilities.
It's a complex problem.
But they're making progress.
Oh, definitely. Lots of exciting research happening.
Good to know. Okay, back to that, true or false. All efflux pumps specifically dedicated to expelling antibiotics.
That would be false.
False, of course.
We've talked about how efflux pumps have all these different roles, right? Expelling toxins, bile salts, metabolites.
Right. Not just those antibiotic bad guys.
Exactly. Essential for bacterial survival in all sorts of environments.
Speaking of those diverse roles, we should mention their impact on bacterial virulence, too.
Oh, absolutely. Some efflux pumps, they can expel those host-derived antimicrobial peptides.
Which are part of our own immune system, right?
Right. Our first line of defense.
So not only are these pumps helping bacteria resist antibiotics, they're also helping them evade our immune system.
A double whammy.
For sure. Okay, one final question for our listener. Ready?
Bring it on.
How can mutations in porin proteins contribute to antibiotic resistance? Think back to our discussion about, the structure of gram-negative bacteria.
Ah, good one. Back to the basics.
While they're thinking, I just want to say, understanding efflux pumps, it's crucial for developing better treatments, right?
Absolutely. Knowledge is power, as they say.
Okay, let's reveal the answer. How can mutations in those porin proteins contribute to resistance?
Well, mutations in porin genes, they can change the porin protein structure.
Makes sense.
And that can lead to, fewer porins, narrower porins, or even no porins at all.
So basically making it even tougher for antibiotics to get in.
Exactly, like reinforcing the castle walls.
Love that analogy. Well, I think we've covered a lot of ground today in our deep dive into efflux pumps.
We have, from the different families to their mechanisms to the challenges they pose.
And we even touched on some promising research avenues.
Always looking for solutions.
Absolutely. Now, I think a poem is in order to wrap things up.
Ooh, I like where this is going.
You always have the best poems.
All right, here goes. In a microscopic realm, a silent war unfolds, where bacteria fight Their secrets they withhold. Efflux pumps stand guard, like soldiers in the night, expelling our weapons with all their tiny might. Antibiotics attack, a relentless, fierce storm, but bacteria resist, their defenses transform. A struggle for survival, a battle yet untold. As resistance evolves, a story to unfold.
Beautiful. It really captures the essence of this ongoing battle. Thank you so much for joining me on this deep dive into the world of efflux pumps and antibiotic resistance. To our listener, thanks for tuning in, and until next time, keep exploring the fascinating world of microbiology.